So one way that you can go and teach monoclonal antibodies to students is that you can go and model it to them, then get students to go and model it back to each other. Make sure that when you're modeling it to them, you've got some nice big models available. Break down each of the uses up into lots and lots of key bits, so it goes and it lowers the cognitive load. And then go and think about what questions you can ask them as you're moving through, so it will really help them understand. The next short video will just show how we can go and model just the use of monoclonal antibodies in pregnancy kits, just to give you a flavour of how to do it. One way that we can go and teach about the use of monoclonal antibodies that comes up on the spec is about how they're used in pregnancy kits. So the first thing that we talk about is the fact that the tip of a pregnancy kit is dipped into urine, right? Just make sure that students are aware this is because the urine is what's going to contain the hormones that the pregnancy kit is going to go and detect. So the second part is that the urine containing the HCG molecules then goes and moves along our pregnancy kit. It might be worth at this point, if you have some chromatography paper and some coloured dye, you could actually go and show students what it actually looks like as this might move along it. Right, the third stage is that the urine is going to go and reaches the mobile antibodies in the strip, right, and not the immobile ones. So we're going to have the mobile antibodies just here, and those growth hormones, those HCGs, are going to go and reach these antibodies. Right, so it's going to go and reach those antibodies over there. Right, at which point the HCGs are going to go and bind to those specific binding sites. So whenever you're talking about monoclonal antibodies, make sure that you keep talking about binding sites. So the HCG will go and bind to those binding sites on those antibodies in the mobile phase. So at the moment we go and we've got antibodies in the mobile phase that are moving up down there, and they're now going to go and attach to the antibodies in the stationary phase. So we go and we talk about the urine containing the pregnancy in the pregnancy test kit going and moving along, and then they will go and bind to these immobile antibodies in our stationary phase, right? As you're doing this, you can almost go and talk about it in terms of almost making a little sandwich of antibodies. So make sure that as you're going along, you can go move things about. That's fine as you're moving along. So now you can go and talk about the fact that the HCG antibody complexes are binding to those immobile enzymes, like we just said, and we've got pairs of enzymes now that are kind of stuck together. The antibodies contain an enzyme at the top end that's going to go and cause a changing colour of a dye, right? And it's that changing colour of the dye that's going to go and cause our pregnancy test kit to go and turn from blue to red, right? So the enzymes that are present there will go and turn the test kit from blue to red if they're present. If there is no of this HCG present, then those enzymes won't go and bind to those immobile antibodies on that binding site, and they will carry on moving along, so you'll have no enzymes present there in order to go and cause a colour change in the dye. After you've gone and you've modelled the concept to the students, you can go and give them a small model for themselves that they can go and work in pairs. They can go and annotate it. Give them some key questions at the bottom just to go and help focus them. After they've gone and annotated it and planned their own short presentation or modelling activity, they can go and model the use of monoclonal antibodies back to another group. Then another group can do it in vice versa. So students will go and model it back to each other.